Okay, so let's hear the talk of Martin from Delft on the structures in parameter space of three-dimensional Turing-like patterns.
Perfect. Thank you so much for the introduction, and many apologies from my side for not joining you in place down there in Marseille, but the corona pandemic just didn't allow us to travel here from the Netherlands. So I have to join you virtually, but thank you so much for the invitation to do so and to talk about this work. I'm going to present a very expository project, sort of early stages here. We're hoping to raise some interesting questions rather than providing answers. And as it was just said, it's about investigation of structures in the parameter space of three-dimensional Turing-like patterns. So I'm going to quickly cover a little bit about Turing patterns, their generalization to 3 dimensions, and then I'm going to tell you a little bit about what we investigated in the parameter space and what interesting questions we came up with from this investigation. I should mention that, yes, I'm at TU Delft in the Computer Graphics and Visualization group, but this work is joint work with Ulrich Reitebuch and Erik Zimmermann, both Freie Universität Berlin in Germany. So let's get started, and let's maybe recall what are Turing patterns. Turing patterns are named after their inventor, Alan Matheson Turing, who came up with the formulation of these in 1952. And he was actually aiming at the description of animal patterns. Think of a cow, a cat, any animal with dots, possibly stripes even. And he was trying to describe how these patterns come to be. There has been work that actually showed that these patterns are relevant in natural development. On the left-hand side here, you see some work by Asai and Rito, who describe the skin patterns of zebrafish in terms of these Turing patterns, as I've just shown you in the formula. But these patterns have more than just applications in skin patterns of animals. For instance, Pringle and Tarnita were able to describe what you see on the right-hand side by Turing-like patterns or Turing patterns, where they analyzed termite colonies, these insects that form these little green patches in the desert and defend it against other colonies. And you can describe these systems by the same equations of Turing patterns. Next on the stage here for our story is David A. Young, who came up with the local activator and inhibitor model for these skin patterns, basically creating the same patterns as Turing described, but with a slightly different formulation. Namely, he's considering 2 different types of morphogens. One activator morphogen that, if it is present in a cell, causes this cell to color or to get a differentiated state, and an inhibitor morphogen that prevents cells to be colored. And the main idea here is that the activator morphogen spreads in a smaller radius, while the inhibitor morphogen spreads on a faster and thus on a wider scale. So you get these isolated pockets, as you see in the leftmost image here. You get these isolated pockets of differentiated cells, and then wide space around them where there is no differentiation. And as you vary these diffusion speeds of activator and inhibitor, you do get different patterns, as you see here on the right-hand side. Oh, by the way, I cannot see the chat, so if anyone's asking questions in between, you just have to stop me and ask them via audio, please. All right, so how does this transfer to cellular automata? Well, the generalization to cellular automata is quite straightforward. You consider your central cell in the right-hand image here. There would be the cell C in the center. And you take one disk around it and you count all differentiated cells, all cells that are activated currently. And all these cells in this small gray disk, they contribute to your central cell C being activated in the next time step. And then you go to an annulus around these cells, and all activated differentiated cells in this annulus are considered to be influenced in terms of the inhibitor, so they contribute a negative value to being for your cell C to be active in the next time step. So simply counting all the A's, all the I's in this image will tell you whether the central cell C will be active or differentiated in the next time step or not. And that's basically what the formula says here. So this is a perfect two-dimensional formulation of these patterns. And here you see some images from a publication of Gary Greenfield, who uses this cellular automata approach to consider what he now says Turing-like patterns, like because they are not differentiable anymore. These are discrete patterns now because they come from this discrete cellular automata. But you see that you basically end up with the same likeness. Here on the right-hand side is the same procedure, just slightly different parameter values for this inner circle radius and the outer radius that then defines this annulus. What you can already see here is that the left-hand image seems to contain quite a lot of almost bended strands. So if you would reduce this by the inner radius, you would get a lot of what we're going to call one-dimensional fibers, while the right-hand side is rather made of these black dots, right? So if you would shrink that, these would rather be like one dot and then around them a little bit of black puffy space. We're going to come back to that in a second, but let me first tell you how to take this formulation to three-dimensional space. So instead of a circle in the center, we simply use a ball. And so all the cells living in this ball will contribute to the central cell being active. And then we're going to use a ball with a cavity around instead of an annulus. So within a larger ball, all the cells that don't lie in the central ball will contribute to the central cell not being differentiated in the next time step. The visualization of these patterns is now more complicated, more involved, sort of impossible, because if we draw all the cells, then you will get these big blobs of cells that obfuscate all the view of what's behind them. So what we decided on how to visualize them here, and this is a picture from an earlier publication from 2017, we decided not to draw all the differentiated cells, but to take regions of differentiated cells and only draw the border to the regions of undifferentiated cells. So let me tell you how to read this picture on the right-hand side here. You will see gray and green regions. If you look at a green spot, it means you're looking through a differentiated region onto a border to a non-differentiated region. If you see gray, you look at the border of an undifferentiated region, which would be a wide or empty maybe, onto something differentiated. So you now do not see these white, not differentiated, black, differentiated points anymore, but you only see the borders in between these blobs. There are actual applications of three-dimensional Turing patterns. I've brought two just to make the point here. On the left-hand side here, you have Tamas Bangasi, who looked at chemical diffusion microemulsions and showed that these do form what can be do form patterns or structures that can be described by Turing patterns. And on the right-hand side here, a physical investigations that also found in these magnetic field settings, also found Turing patterns, three-dimensional Turing patterns. So just like their two-dimensional counterparts, three-dimensional Turing patterns are actually a thing out there in nature. So it's interesting to study them. I was told that this animation here runs very slow for you, but don't worry, it's not so much important that you see all the aspects of this animation. What you should see right now is that from a sort of random state, and this randomness is this factor rho here. So rho tells you that in the initial time step, there is about a 50% or actual 50% probability for a cell to be differentiated or not. Then we have an inner radius of 4 and outer radius of 5 here. And you see that after a couple of time steps, I'm showing 20 here, certain patterns form, and this image on the right-hand side consolidates to a structure. What is important is not so much that you see what's happening here, but that you see the difference to the next animation, where we vary the inner and outer radii. And hopefully you do see that there are still structures forming, but these structures are not as fine-grained as they were before, but are of way larger scale, which is to be expected if we choose way larger radii. A couple of interesting things to be considered here is how fast this converges. So this is actually something that can be feasible in an experimental setup, choosing a couple of parameters and then investigating the outcome. So in this 2017 publication that I've shown to you, we were able to find a set of parameters that gives us these little balls. So in this case, those are undifferentiated balls. They live in very separated in space. We were also able, with a different set of parameters, to create a structure that has some tube-like behavior. So you can actually dive into one of these tunnels and dive through the entire space that is covered here. So in a sense, on the left-hand side, you see balls that if you shrink them by the inner radius, just become points, zero-dimensional objects. And on the right-hand side, you have these tunnels that bifurcate and meet again. But if you shrink the tunnel by the inner radius, you will still have the bifurcation. So you will have, in a sense, a one-dimensional network. And this does recreate what we saw in the two-dimensional case, where we had blobs or one-dimensional strands. But now we moved into 3 dimensions. So the question that we asked in that 2017 publication is, what about two-dimensional structures? Can we find those? Can we find a parameter set that sets the parameters to two-dimensional things that, even if you shrink them by the inner radius, still have a two-dimensional extension? So we discretized this parameter space. We went with the inner and outer radius. We went from 1 to 40. In the probability of being activated, we chose 120 or 121 rather different values, and they are not linearly distributed between 0 and 1, because we found in prior investigations that the interesting cases happen rather at those spots where it's very low or very high probability of being activated. So the activation probability is distributed among the sigmoid curve here. And what we did find is indeed 5 different or qualitatively different types of structures. On the one hand side, these differentiated spheres that you've seen before in the image, then some differentiated pipes. So the differentiated cells make the inner parts of the tunnel, and the outer part is dead cells or non-differentiated cells. We found the corresponding counterpart. So this is just a dead volume, an inhibitor volume, an undifferentiated volume surrounded by all differentiated space. And these undifferentiated tunnels or pipes that are surrounded by living and differentiated patterns. But we also found these two-dimensional extent structures that clearly, if you shrink them, still have these clear two-dimensional extension. So here's a visualization of the parameter space. Here's the inner radius, the outer radius, and the on the third axis you have the activation probability. And you see this this plane drawn into the parameter space. This is the plane where the volume of the ball covered for the activator and this ball with a cavity for the inhibitor is equal. And you also see that not so much interesting stuff is happening above the plane. Most of the points here above the plane would be those settings where everything becomes undifferentiated, so we don't show them here. Here's a histogram of all the structures that we found. So you see this is a log scale, so these differ quite a bit. Most of the structures we found are actually completely undifferentiated, so all the cells in our domain are just undifferentiated. The second place is taken by everything is differentiated, which is the other biggest probability. And then going down here, we have differentiated cells, differentiated tunnels or pipes, areas, undifferentiated pipes, and coming in last are the undifferentiated spheres. So here's the same parameter space. This black part is what wasn't shown before, and now you see why, because it would black and obfuscate the entire view on everything behind it. So this is most of the space is just covered with dead cells. So however you start from these parameters indicated here, you just get dead volume. The other biggest part are these white ones here. These are the all is differentiated cells. And then the two other big parts are here, the differentiated spheres, and here the differentiated pipes. And on the left-hand side here in this image, I show you isosurfaces between these elements. And you can see that there's a little wedge rising between this yellowish beige surface and the green one on top. So it's sort of triangularly opening, which does enable our first conjecture. Namely, we assume that this opening is actually getting larger the further you go out. So if we would go increase our maximum radius radii, we would get a larger space there or a larger volume. These area UC pipes and UC sphere structures only arise around this hyperplane that I've shown you before, but we still do conjecture that if you go far enough out with your maximum radius R1 or R2, these will also actually gain volumetric extent. Volumetric not in the sense of the Turing pattern, but in the sense of their corresponding parameter sets in the parameter space. So one thing I haven't told you, and let me take maybe the last 2 minutes to do so, is how we did classify these. So if we did pick one element, one of these points here in parameter space, how did we give them their color. And this was done purely manually in a citizen science experiment, actually, where the citizen scientist chose a parameter set. They had a simulation run. They were presented with the corresponding Turing-like pattern, and they chose which of these 7 possible cases they saw. So one interesting and maybe final question for this talk is, how would you automatically classify these patterns? And one thing we looked at here, and this is our first step towards automatic classification, is we plotted some of their values. So the color comes from the qualitatively and manual labeling by the citizen scientists. Here you see a plot of the volume versus the surface of these structures. This is not so interesting. Maybe more insightful here is the plot of the volume against the activator radius, which both of these do suggest that the volume is sort of a very good predictor of what structure you have here. And indeed, if you print these as a violin plot, you see that the DC spheres, the DC pipes, the areas, the UC pipes, and the UC spheres are pretty well separated along this volumetric axis here. So the one thing we did, and that's the final experiment we did, is we took 5 folds of our entire data set, and we trained a k-nearest neighbor classifier on the remaining points and tested it on the respective folds. And you see that in total, we do get, on the bottom row here, we do get very reliable classification results, but you also see that the misclassification varies drastically across these different types of structures. So there's still work to do in figuring out how to classify these structures automatically. And finally, I'll leave you with this conjecture. Namely, we looked at 3-dimensional Turing patterns, and we saw 0-dimensional structures in 2 flavors, A and E in this picture. We saw sort of 1-dimensional strands, tunnels, pipe structures in 2 flavors, B and D. And we saw areas in obviously 1 flavor, because flipping the coin here doesn't make so much sense. And so we conjecture that if you go to higher dimensional spaces, then say into D-dimensional Turing-like patterns, you would see all these structures all the way up to D-1 dimensions, where all but the D-1 dimensional ones appear in these 2 different flavors, differentiated and undifferentiated. And yeah, that was all for my talk. Thank you so much for your attention. And I'm happy to take any questions you might have at this point. Thank you.
Thank you. Other questions? Right. So Nazim has one in the chat. I will leave you to read it because.
So it says, a small historical remark about Turing's 1952 article. He was not looking for how patterns appear in animals, but rather he was interested by the problem of morphogenesis, that is how shapes appear in a symmetric environment. Yeah, that is very true. That's the title, actually. Yeah. So to reformulate it with a modern vocabulary, maybe we could say that Turing was interested in a problem of symmetry breaking. Of course, as a byproduct, Turing was happy to observe that his model reproduced the patterns observed in various animals. Sure. Yeah. Thank you for that. That's a good clarification, actually. Turing was not after understanding how these patterns emerge, but rather after this question of morphogenesis. But now we do have this great tool, and we did find out that it does describe skin patterns. So it's a good opportunity for us to apply this tool, or this toolkit rather, to different scenarios, as I've shown, like in physics or chemistry even.
Okay, I'll ask a question. If you run your 3D CA and then you project in 2D, do you get something that is still Turing-like? And I mean, if you project class, I don't know, class B, do you obtain a class B? Or I mean, is there something that you can say about how this goes under projection?
Yeah, that's a really interesting question. So what you could do is you could take your three-dimensional pattern and you could slice it, right? Because each horizontal slice will now give you a two-dimensional pattern, and any axis parallel slice will actually give you a Turing-like pattern for the corresponding inner and outer radius that is stable and wouldn't converge any further. I am not sure actually right now what happens for non-axis parallel slices, but I would think that the general statement still holds. So independent of how you place like an affine plane in this three-dimensional pattern, and you just slice it with that, you should always get a two-dimensional, or well, if you go to the higher dimensional cases, a corresponding lower dimensional Turing-like pattern. And there's another question in the chat. Are sponges possibly produced by three-dimensional reaction diffusion phenomena? This is a super fun question. I don't know, but I guess one should investigate that, because yeah, once you state that, I guess you can't unsee that in particular these pipe-like structures do have quite a similarity there. I don't know, is there a sponge expert there? Okay, so apparently there's a source in the chat that says so. Perfect. Okay, yeah, so sponges do. Good thing we answered that.
Hi, Martin. Great talk.
Thanks.
I was just wondering, you mentioned at the beginning about the Turing and Young having different answers to the same problem. Are these like fundamentally different approaches, or are they just kind of slightly refined versions of the same idea?
They are, if you go to the differential equations, they are slightly different in the sense that they do use a different weighting scheme. So as far as I'm aware, Turing basically said there's this one morphogene passing around, and that will tell you whether a cell is differentiated or not. And the qualitative difference to Young's approach is that there's a second morphogene now, and these two are interacting. And depending on the weighting that you give to the one and the other morphogene, you can get something that is exactly Turing's definition, but you could weight these differently, and then you will get another system, a system that is possibly capable of creating different structures, say. But we didn't look into that too much. We focused on this one formulation that does give you the Turing system.
Thanks.
Yeah, welcome.
I have a question, but I didn't understand quite well. To maybe this is something not very interesting. So the definition of whatever definition you have for Turing pattern or Turing-like pattern,
yeah,
is it? I guess in the setting of cellular automata, it doesn't make sense. But for original Turing patterns, are they invariant under rotation? So if you have a Turing pattern and you rotate it, do you get another Turing pattern?
So the Turing pattern, the original formulation of Turing is completely symmetric. So if you go to his paper, the very first formulation is on a ring of cells, and all these cells are completely symmetric. So if you fix a state, a converged state, and you rotate that state, nothing changes. You just get like a different converged state, and that holds also for the two-dimensional generalization. And so it does for the one of Young and the cellular automaton. So even if you take the converged cellular automaton and you translate it, you shift it, you rotate it, you always maintain the converged stage and get yet another pattern by the operation you applied.
So for cellular automata rotation, and what exactly do you mean? Is it rotation?
So if you imagine like your two-dimensional converged state, and you rotate it by 90 degrees, so each cell goes to, I don't know, around the center of your domain where you apply this, each cell goes to like a 90-degree rotated version of itself, then this is still in a converged state and will still give you a converged two-dimensional Turing-like pattern.
So is it rotation under 90 degrees? Or
yeah, of course, because sorry, I should have said that. So obviously you need to have some operation that maps your discrete cell grid onto itself, but under each of these operations it's a converged state maps to a converged state.
Okay, but for Turing, the original Turing patterns in two-dimension rotation, they are invariant under rotation in any with any angle, right?
Yeah, exactly, because these are differentiable formulations. So you can actually take this entire continuous domain and rotate it at any angle, and you will get a different Turing pattern. Yes, you're right.
Okay, thank you.
Yeah, you're very welcome.
Okay, thank you very much. We need to move on to the next talk, but thanks again.
Yeah, thank you.